I really appreciate the opportunity to give a talk and just to kind of indulge my curiosity. How many of you have heard of nanodiamonds before? Oh, great. Fantastic. So the literature is working. Okay, I just wanted to be sure. Okay. So what I hope to show today is that diamond is renowned for its beauty, for its facets, a lot of applications, but also it's very multifaceted in its application in a number of very high impact industries. Now, of course, I won't cover all of these today, and I'm going to limit the focus to the applications on the left as they relate to biological and medical applications. I think it's important first to kind of give a general classification of diamond. There's generally two types of diamond, type 1 and type 2. Type 1 diamond is notable for having nitrogen impurities as its primary impurity constituent. And as you will soon see, that the nitrogen is the most important player in this game of color centers in diamond. Type 2 diamond, which I won't really discuss, contains almost no nitrogen impurities. And then if we then consider natural and synthetic diamond, we have terrestrial diamond, which is diamond that forms in the earth, and impact diamond, which can form from meteorite impacts, can be found in craters, for example, Krasnoyarsk and Tunguska in Siberia. Synthetic diamond, in general, three major classifications: high pressure, high temperature, detonation nanodiamond, and chemical vapor deposition. For this talk, I'm really going to focus on HPHT and detonation. Mostly HPHT, as this is the one that's used for fluorescent nanodiamond production. These two are the most important for this talk. Okay, so I wanted to give a brief short anecdote about fluorescence in diamonds. And one of the most famous diamonds that exhibits a very unique luminescence is the Hope diamond, which is in the Smithsonian. And there is a very interesting history of this diamond. Supposedly it was stolen out of the eye of some statue, and then some religious statue, and then the priest then cursed it. So there's this long lineage of everyone who's come into contact with this diamond somehow mysteriously dying or encountering some type of calamity. And that includes Marie Antoinette, all the way down to the postman who actually delivered it to the Smithsonian. I don't remember what year it was, but it was shipped for $2.55 to the Smithsonian with $155 of insurance placed on it. So even the postman was not excluded from this apparent curse. But actually, and I suppose if you showed any of these victims of this diamond, this blood red fluorescence, they would say, aha, I knew something was up with this diamond. But as it turns out, it's really just an effective boron and nitrogen impurities that give this really intense red phosphorescence. Although I suppose that doesn't exclude it from still being cursed. But anyways, in general, there are hundreds of optical color centers in diamond. And for the talk today, I'm really just going to focus on really these NVN and NV center. And all of these optical defects are caused by impurity atoms and structural defects within the diamond lattice. Three important ones which involve nitrogen and vacancies. And vacancies are just simply point defects within the lattice, where there should be a carbon atom and there's not. So this N3 center, which provides blue emission, the NVN center, which provides green emission, and the NV center, which provides red near IR emission. And again, for this talk, I'm really going to focus mostly on the NVN and NV center, as these are the most established currently commercially. How do we actually produce color centers in diamond? Well, first we need to introduce vacancies, which is then accomplished by irradiating particulate diamond or bulk diamond with high energy irradiation, be it electrons or neutrons or protons or what have you. We then heat treat this diamond at high temperature. Vacancies diffuse, they move around in the lattice and then form complexes with the nitrogen, which gives you your color center. And then we fragment these diamonds down to the nanoscale. And there you go, we have our fluorescent nanodiamonds. I would like to briefly just highlight a recently developed technique. Our group, as well as collaborators in Italy and City University, New York, we developed a new technique of annealing, which shows that you can actually control these color centers in synthetic diamonds. So heat treating at various high temperatures very quickly can lead to these very interesting fluorescence, not interesting and novel fluorescence in synthetic diamonds. So we can create these 8 NVN centers, mixtures of green and red emitting centers, and it's the N3 center, which gives you this blue luminescence. It's enough of the solid state kind of physics, and I will go into, and I want to kind of discuss the particular features, characteristic fluorescence features of diamond, particularly if you're using it in some biological application. So for the NVN center, peak absorption is around 480 nanometers, with peak emission being around 520. For the NV center, peak absorption is around 575, with peak emission around 680 to 700 nanometers. And I wanted to give a brief comparison, the quantum yield, and to a very commonly used dye, Alexa 647, which is pretty much a workhorse dye in a lot of applications. So you can see the quantum yield for color centers in diamond is around 0.7. For Alexa 647, it's about 0.33. And then for fluorescence lifetime is about an order of magnitude roughly higher for color centers in diamond as compared to this Alexa 647. So they're very suitable for these lifetime time-gated imaging applications. And just to kind of orient you with size, currently nanodiamonds are optimized kind of in fluorescence to this 40 to 100 nanometer range. There are smaller particles, but down to 10 nanometer, but they have some other additional applications, not directly just related to contrast imaging in cells. So 140 nanometer particle is about approximately the same brightness as a single quantum dot. And I did want to just reemphasize the nature of this fluorescence. So it's not that the entire particle itself is fluorescent. The particle has individual fluorescent color centers packed within this single particle, which all fluoresce. And then you get your fluorescence from the particle in that way. Quantum dots or polymer dots then emit kind of uniformly, and then fluorescent, for example, then emitting as this molecular fluorescence. So the key features of diamond, and perhaps one of the most important feature of the fluorescence of diamond color centers, are their infinite photostability. And I showed 2 examples here. This example was demonstrated by Reineck and coworkers in 2016. So this was our group recently. So what we do is we had blood clots which were labeled with fluorescent nanodiamond, and another set which was labeled with phycoerythrin. Yeah, it's another commonly used dye. So phycoerythrin is actually notable for its photostability issues. But anyways, you can see that after 30 seconds, this phycoerythrin is then almost not visible. It's photo bleached completely. In the case of Alexa 647, same, after 40 seconds, you can see it's effectively bleached, but our diamonds containing NV centers are still present. And then just a general comparison of fluorescence versus time compared to several other commonly used or available fluorescent materials also shows similar decay, but of course, the NV center is stable with time. Okay, so we have photostability. The next key feature is biocompatibility. And suffice to say, there is a profound amount of literature out there which has demonstrated that diamonds are quite biocompatible. In general, I wanted to just list these here. These are a little more recent. So this has been demonstrated to be biocompatible in zebrafish, rats, primates. I believe there was also a canine study. And then there's a couple reviews here which address this biocompatibility as well. These 2 examples here are related to our research. These are endothelial colony forming cells, which demonstrate that effectively the cell viability does not decrease with increasing dose of nanodiamonds up to 100 and even up to 128 micrograms per mil. And in here it's a picomolar concentration, but again, these are in a different cell line. These are kidney-related cells, these 293 KDR cells. This was done by our collaborators at CIBTech, and this was done by our collaborators at Creative Scientists. They're in Raleigh, they're in Massachusetts. But anyways, similar results. So there is no appreciable decrease in the cell viability upon exposure to increasing concentrations of diamonds. So they're quite biocompatible. A third key feature is the functional surface chemistry. So in general, we have a very diverse scheme of surface chemistry we can do with diamond, ranging from just traditional organic to biological species. Any click chemistry is available. Grafting with polymers is available, streptavidin antibodies, and then biotin PEG. And in general, this quick example here, I just wanted to demonstrate, we took 1 micron red fluorescent diamond particles and 1 micron green fluorescent particles. So the green fluorescent particles were functionalized with biotin, the red fluorescent particles were functionalized with streptavidin, and we just mixed them together. And then you see this aggregation, which is really the demonstration of this interaction between biotin and streptavidin over time. So it's a simplistic demonstration, but it is a demonstration that you can functionalize the surface of these particles and not interfere with the functional capability of the molecule, for example, the streptavidin. So a fourth key feature is that they are quite chemically robust. So they can withstand very harsh chemical conditions. And in fact, what this example is showing is they can withstand this tissue digestion. So you can upload or introduce particles in vivo, and then ex vivo organ analysis or tissue digestion can then allow you to see the amounts of diamond that the fate of these particles in vivo. So it's quite an interesting application. And then this fluorescence spectroscopy is kind of undeniable. That is 100% the diamond spectra. So it's quite an interesting application for allowing for a quantification and determination of fate of the particles. And lastly, I will touch on this briefly in some additional application slides, but the spin properties of the NV Center, since it has a free electron spin, you can actually, it is sensitive then to these external applied magnetic fields, microwaves, electric fields, and it's been demonstrated even intracellular temperature, which I will show on a later slide. And you can also have the ability to then modulate this fluorescence with these external fields. And this is applications in not only imaging, but also in nuclear magnetic resonance and MRI. The next several slides, I want to go through just several applications and demonstrations of diamond as they relate to biological applications specifically. So I'll start mostly whole body imaging and targeting with VEGF is the work that our group and collaborators did over the past couple years under a phase 1 and phase 2 contract from NIH. The primary goal of our research in this endeavor was to use the diamonds to label tumors in vivo and then to image the particles in vivo. So the question that we initially had was, well, can you actually visualize these particles in vivo with whole body imaging? And that actually hadn't been done before, especially with an intravenous injection. It had been done with subcutaneous injections, so just injections under the skin in mice, but not just administered in vivo via intravenous injection. So we took these mice and injected them through the tail vein with increasing concentrations of approximately 170 nanometer particles and imaged them at IVIS, and this was done at Duke University. And with the IVIS system is an in vivo imaging system. Excitation, these are showing just 2 different excitations, 605 and 640, and we see accumulation in really mostly the spleen and liver, which is kind of expected because those are the filtering organs, right? So you do see this increased signal versus the control mice with no diamonds. And this is, again, that figure because we ended up doing this tissue digestion with these spleens, actually, and this is where you can see the increasing administration dose and then increasing obvious concentration of the particles in the digested tissue. So then the conclusion is that, yes, okay, we can see the diamond particles in vivo with whole body imaging. So then the next step is then to see, well, I should add, these mice do not have tumors on them yet. So the next step we wanted to do, since we want to target tumors, is to then introduce tumors into the mice. This application but we first don't want to use targeting. So this application is going to rely on this EPR, enhanced permeation or tension effect, which is essentially just, okay, it's this non-specific uptake of particles that tumors will exhibit because they kind of just consume everything around them. So anything going around them will kind of get taken up by this tumor. So this is kind of what I would say non-specific. So it's not targeted. And what we did end up seeing, so these are non-targeted diamonds, but injected into mice with tumors. And we can actually see with ex vivo analysis, and this was done with IVIS as well. So this is just taking out the organ and looking at its fluorescence emission intensity. So we do see versus a control, which is just a dextrose buffer. We do see an increase in the fluorescent emission intensity of the tumor and then the spleen and liver. So there is some merit, obviously, to this EPR effect, and it potentially could be used to label tumors. However, EPR in and of itself is quite heterogeneous. So it's not an ideal technique to use to label tumors. So the next step is then to target the tumor directly. But first we needed to develop a targeting vehicle and a scheme to do that. And what we chose was to use VEGF, or vascular endothelial growth factor. And VEGF is essentially a class of proteins which stimulate vasculature growth. And they're involved not only with tumors, but also in, you might imagine, wound healing, things of that sort, and tissue growth. So essentially what happens with a tumor is that these cancerous cells rapidly need oxygen. They're oxygen starved. So this causes tissue necrosis surrounding them. So what they do is there's a feedback loop which then drives these cells to overexpress VEGFR receptors on their surface. So first they start to output more VEGF protein itself, and then a feedback loop then causes these cells to overexpress these receptors. And then that stimulates this angiogenic kind of rampant vasculature growth, which feeds the tumor and continues its growth. But the thing is, if we can functionalize our diamonds with VEGF, we can exploit that as a means to target these overexpressing cells, so these tumor cells. So we did that via a click chemistry route, and I will not go into extensive details of that, but it's essentially relies on transcyclooctane tetrazine chemistry. So it's just bioorthogonal click chemistry to attach the VEGF protein, single chain VEGF protein, to the surface of the diamonds. And we did several validations to verify that the VEGF was attached and functional. Just ELISA and SDS page, ELISA is really a plate-based technique. And then the SDS page. In general, we can see that the VEGF is functionally active, is functional, and it's on the particles. So our conjugation scheme does work. And in general, it's also not just non-specific uptake of the VEGF, it is covalently bound. So the click chemistry works. And then there's this tyrosine autophosphorylation, sorry. I'm not going into details about how that works, but in general, it then demonstrates that these VEGF functionalized diamonds activate this autophosphorylation pathway at similar concentrations to free single chain VEGF at 0 nanomolar concentration. So, great. We can see that the VEGF is on the surface, so our conjugation scheme works. And the next slide, I just want to go through this competition assay. So this competition assay involves using a toxic variant of the VEGF protein. So essentially what happens is when this toxic variant binds to the VEGF receptor on the cell, it then will cause the cell to die. If we then mix this toxic variant protein with our diamond, which contained VEGF, they will compete for access to these receptor sites on the cells. In general, if the cells can then survive, we demonstrate that the VEGF on the diamond is active and successfully can compete with the toxic variant of the VEGF. And in general, we do see that. So it is comparable to just a free VEGF variant, single chain VEGF. Okay, so I will say that the IVUS imaging of these targeted examples was not as good, I would say similar to the EPR-based approach. So, and really that just needs optimization. But the ex vivo analysis of the tissue is pretty unmistakable. So this is the control. So this is no FNDs administered. This is FNDs administered, but not containing VEGF. And this is FNDs with VEGF, which were administered. So it's very obvious that the diamonds are accumulated in the tissue when they are targeted to VEGF. And then we actually then normalizing this to tumor area, we can see that the uptake is clearly, the fluorescent signal is clearly higher in the VEGF variant of diamond. So in general, right. So this ex vivo analysis is excellent, and it does validate that the diamonds do work in that sense, and they can be visualized. And in general, this work is recently published in our bioconjugate chemistry down here. Okay, so that was essentially our work, and I wanted to go through some very famous kind of examples in literature of some other demonstrations of diamonds. So this was a pretty seminal paper of using diamonds to target tumors, and this is actually using detonation nanodiamonds, so not fluorescent. But actually what they did was upload doxorubicin, which is a chemotherapeutic drug, which is used to treat cancer. And what they found was that loading this drug onto the diamond surface causes a reduction in the tumor growth, right? So the basic concept is that these small particles have very high surface area, so you can upload a lot of drug onto their surface and then get this slower, controlled release of drugs directly at the tumor, as opposed to just a free drug, which then is very fast diffusion and can almost, you need to administer much higher doses actually of this free drug variant, which is really a problem. Chemotherapy is not a very good treatment in terms of the patient. It's very unfortunate because these are very toxic treatments. So this approach can allow you to minimize the dose, but get, in effect, better efficacy of the drug. Okay, so moving on to just a couple imaging applications. This paper is currently our collaborators and myself actually wrote, and this was utilizing this rapid thermal annealing approach. So we were doing this 2-color imaging with green and red fluorescent diamonds. The green fluorescent diamonds were produced in rapid thermal annealing approach. And you can see this 2-color, these are in breast cancer cells. So we took basically 2 different lines of the same breast cancer cells, put green diamond in one, put red diamond, and then mix the cells to just demonstrate. So these are not targeted in any way. It's just non-specific uptake to the cells. But okay, we can see this 2-color imaging, and there have been some other demonstrations of 2-color, but this is one of the first ones where it's actually been done with confocal microscopy and not with some electron beam excitation or cathodoluminescent microscopy. Or in one case, they're using IR emitting and NV emitting. So this is green and red emission from the NVN and NV centers. And I'd also like to point out, I don't show them here, but there's also the possibility of producing these yellow fluorescent diamonds, which are combinations of NV and NVN centers in the same particle. And as a result of this green emission from the NVN center and red emission from the NV center, you get yellow emission. In general, these can have some very interesting potential applications that we're thinking about with this fluorescence modulation. It would be interesting if there's some energy transfer between these 2 different color centers or how they interact with each other. But so that manuscript was just submitted, and it's actually, we're responding to reviewer comments now. So hopefully that will be published soon. And mentioning the utilization of the spin properties, I just wanted to, these are kind of also seminal papers where this is somewhat demonstrated. So this paper demonstrating that you can use microwaves to modulate the fluorescent signal of the NV center with time. And in general, what this allows you to do is to get background-free imaging. If you have tissue autofluorescence in the background, you can then modulate your fluorescent signal of the diamond particles and you get much higher confidence in what you're looking at, essentially. And then in addition to microwave modulation, this magnetic modulation demonstrated in 2014 also allows you to enhance the visibility. So these are lymph nodes with diamond, and then magnetic modulation allows you to enhance your contrast. I wanted to go into kind of a very interesting application, other application where this concept of the doxorubicin example, this is instead not doxorubicin, but amoxicillin, which is a common antibiotic. And this was actually done, Dean Ho's group at UCLA. They've done a lot of, that was also the doxorubicin work was his group too. He's currently at National University of Singapore. But so essentially, gutta percha is a polymer that is used in root canal implants. But they often have trouble with their mechanical stability and the likelihood of reinfection. So what this paper did was introduce diamonds with amoxicillin on their surface. So the amoxicillin provides you a broad spectrum antibiotic protection, so it prevents instances of reinfection. And additionally, the diamond particles provide you a mechanical stability of the gutta percha. And currently this is in clinical trials. I was looking yesterday, supposedly the clinical trials are supposed to end in 2020. So yeah, this is a pretty neat application. It's also showing this approach of having a drug slowly released from the surface of a particle. And last couple of slides, I just wanted to demonstrate the possibility of sensing applications of diamond. So intracellular sensing, in this example, they're measuring temperature fluctuations within the cell. So essentially what this is showing is you have gold nanoparticles, which they then hit with a laser, they radiate heat. and you have a local, an NV center or a diamond containing NV centers at here and at slightly larger distances. And you can actually measure the temperature change. And of course, it's a function of distance away from the spot which you're heating and in general here. But you get this 1/10 of a Kelvin or so sensitivity within the cell. So it's quite interesting. In addition to just temperature sensing, there's the potential to sense electric fields. So for example, neurons firing, for example. In general, these applications are all kind of emerging, and and everything needs optimization, but it's a very interesting perspective for Diamond. And the last application, and this has become quite popular recently. So it's effectively using the spins or the ability to polarize NV centers to a very high degree at room temperature. And then you can then transfer the spin to external molecules, or for example, in this case, this group at Berkeley, Ashok Ajoy, and they do polarize the NV center and transfer that spin to carbon-13 within the diamond lattice, and then the carbon-13 polarizes external nuclei. So in general, what this can allow you to do is local improve. In the case of NMR, it's improved sensitivity. And in the case of MRI, in theory, you can use much lower magnetic field strengths to get the same benefit. And ideally, that will hopefully be an application that will come to light, especially in point-of-care therapy and smaller hospitals, for example, which can't afford a multi-million-dollar MRI machine, right? I just wanted to, again, acknowledge all of our collaborators, as well as Olga, Marco, a lot of collaborators at Duke that we did a lot of this work with, as well as a creative scientist in CivTech CivTech has developed the actual VEGF that we used. And then Philip is really great in the fluorescence characterization of diamond, and Niraj, they were involved with the recent paper on the multicolor imaging of diamond. So, and of course, support from NIH, currently currently we have an NCI project. The phase 2 SBIR ended 2017. And so we currently have this NCI phase 1 SBIR contract. So I just wanted to give you, again, a brief overview of, okay, what are the commercially available if I wanted to use them in my research. And in general, like I said, we offer sizes from 10 nanometer up to 150 micron. For fluorescent applications, unless you're doing some spin related applications, probably the 40 to 100 nanometer is right now where things are optimized. And we hope to, in the future, as the techniques of synthesis and manufacturing improve, we can hopefully get down to these very small, ideally sub 10 nanometer size scales, but that will take some time. But it's a big technical challenge, so those things take time. Again, I won't go through every aspect of the slide, but it's here. So we're available and we sell a lot of these products. So if you're interested, we will definitely be happy to collaborate and offer material for use. And lastly, I just wanted to conclude with, again, that diamond is indeed multifaceted, I think, and its beauty is not only from the facets which impart this beautiful shimmer, but also from the number of applications and very high impact technology implications. So I think I would pose the question to anyone in the room of, where do you see diamond? If it can be used in your research, we're happy to open any type of collaboration. It's a great opportunity if you're interested in using diamond in any way. That will wrap things up. Thank you.